All right, so chapter 6 then, shall we? No, it's not very good yet. Those are my markers that are on probation. Get demoted to the trash pretty soon. So chapter 6 is about viruses. And this chapter, we could talk about viruses for a whole semester and more, right? So the general idea is just to give you a brief introduction to viruses. Notice that it's chapter 6, which if we were doing the chapters in sequence, the context of your authors is that we teach you a little bit about bacteria, remind you some things about eukaryotes, then we talk about viruses, then we talk about, how all these things can cause infections and disease, right? I like to do the virus chapter after we've done chapter 9 because there's a lot of molecular biology in viruses. And so if we had a review, which we just did, of replication, transcription, translation, it helps this go a little bit smoother and maybe gives you guys a little bit more understanding about viruses. So if you take a look at that paper that I gave you, at the top of it is, at the top of it, I believe it says something about general characteristics of viruses, right? And I'm not going to rewrite all of these because they're right there in front of you, right? But briefly, we refer to them as obligate intracellular parasites. Because they cannot survive very long outside their host. So what that means, obligate, must have, intracellular, inside somebody else's cell, right? And they can't survive very long, if at all, outside the host. Different viruses can survive for different amount of time outside the host, depending on whether they're enveloped or what kind of protein caps that they have, right? But they are technically, smell that today, technically not living things. Because 2 of the characteristics of living things that we hammered into you in Bio 1 are you have to be able to produce energy, ATP, some sort of metabolism. Viruses don't do that. That's kind of a big one. And you also have to, in order to do that, you need enzymes, right? And viruses bring some of their own enzymes, but they mostly use the enzymes of the host. Okay, so they don't bring any of their own machinery. They bring nucleic acid and occasionally an enzyme, but they don't bring ribosomes, endoplasmic reticulum, ATP, any of that stuff. So they're technically not living things. They are specific to their hosts within reason. Okay, so there's probably a line on there that says, there are viruses for all types of organisms. So there are viruses that infect plants, viruses that infect animals, obviously we know that, right? Viruses that infect fungus, viruses that infect all the protists, and viruses that infect bacteria. Since we are in microbiology, we're going to spend our time talking about the viruses that infect bacteria and the viruses that infect animals. Right? We're not going to talk about plant viruses. Go to botany and learn all about it. Right? Except to say, of course, tobacco mosaic virus was the first virus that was ever purified and identified. And as its name suggests, tobacco mosaic virus, it causes the leaves of tobacco plants to decolorize in areas like a random mosaic. Right? So that's why it's called TMV. Right? They have to have some sort of genome. And what's different about viruses than any other cells that we've talked about so far is some viruses have double-stranded DNA as their genome, which is what we're familiar with, 5 prime, 3 prime, double helix, complementary bases, blah, blah, blah. Other viruses have just one single strand of DNA. Other viruses, as most of us know now and are more aware of, they can come with a single strand of RNA. Be aware that RNA can either be what we call plus strand or a minus strand. And those are replicated differently inside the cell or multiplied differently inside the cell. There's different steps need to happen in order for that virus to make a copy of its genome and to make proteins. And then there are viruses that have multiple strands of RNA for genome. So I'm thinking about HIV virus, it has 2 strands of RNA. Influenza, I think, has 7, right? That's so it depends on the virus. Each one of these different types of genomes goes through a different step or different steps process of multiplication inside animal cells or plant cells. We aren't going to be able to talk about all of them, right? Because literally each of these has maybe, a 4 to 8 step process. So we're going to talk in generalities, okay? Probably I'm going to focus on double-stranded DNA viruses because there are a lot of those, and probably a little bit about some multiple-strand RNA viruses. In addition to having genome, all viruses have some type of a protein coat. We refer to it as a capsid. It is usually very highly organized, which I'll show you some pictures of later. Like you can do X-ray crystallography to the capsids of viruses and see what their shape is, what they look like. Some viruses, so it's usually, wow, what's that? Some also have an envelope. And an envelope is often a combination of protein and phospholipid, because usually viruses get their envelope from the cells they live in when they leave. So it may have lots of things in it chemically. It's not just protein, it's not just phospholipid, it's not just carbohydrates. Unlike like the glycocalyx, which is a little simpler, right? Or a capsule. And in that envelope or protruding from that envelope, then those would be the spike proteins that we hear so much about with infection, because the spike proteins are literally, like they sound, they're proteins that stick out like spikes, like think, medieval times, pitchfork, pointy stick, right? And those are sticking out, and they almost always, there's a receptor on the host cell that that spike protein will fit into. And the host cell is tricked into thinking, oh, this is something binding my whatever receptor, I must need this, and the cell brings the virus in, okay? So these spike proteins, that's why everyone's so interested in them, because they serve as the mechanism for the way the virus enters most animal cells. Bacterial viruses don't have spike proteins. They're a lot simpler, as you might guess. If I hit all those high points, yes. Host cell specific, usually can infect more than one type of species. Now, this is, it's like with enzymes, right? We say enzymes, one enzyme, one substrate. And we teach you that in BIO1. And in 7th grade, we say it's a lock and key, and there's only one key for each lock. Yeah, well, that's not entirely true. Right? So as you progress along your studies, you'll find that these sort of maxims or, absolutes, sometimes there's a little waffle room there. And this is an example of that, right? So viruses usually do not cross species barriers unless all the species are living in the same environment and are exchanging usually some sorts of bodily fluids, right? So yesterday when I was driving home, I think it was yesterday. I don't know, sometime I was in the car yesterday. I was listening to NPR and they were talking about all these research studies that are coming out that are being published about coronavirus. and how it's most likely derived from an animal virus and most likely from a marketplace, because places like where there are there's a lot of literally blood, guts, organic material, blood, guts, sputum, and not a lot of hand washing. That's a great place for viruses to jump species, right? Flu, influenza is a classic species jumper. Flu lives in pigs, birds, and people, right? That's why we call it the swine flu sometimes and the bird flu or the avian flu other times. What that means is it happens that year, it happens to have more of the RNA, more of the strands of RNA from bird flu than from people flu / pig flu. The mixing vessel in that case is the pig, usually. And it's because it comes from places where people, pigs, and waterfowl all live together without a lot of sanitation, right? So again, a lot of exchange of feces and saliva and breaks in the skin, all different ways that viruses can enter the body, right? You're never going to be infected with a plant virus because that's a little bit too evolutionarily distant from you. You don't share a lot of receptors on the surface of your cells with plants anymore, right? Can you get a virus from another animal? Maybe. Depends on the animal, depends on the virus. Are you picking up viruses from protists and bacteria? No. They're, again, too evolutionarily distinct from you and usually don't have the capacity to enter into your cells. Okay? And if they do get in, your cells have defenses to get rid of them very quickly. So, okay, enough about the general characteristics. Let's talk a little bit about the classification. Oh, I see size. I put size on there, very small. Yeah, okay. I'll put that on there too. Size. Man, we measure them in nanometers. All right, so how do we classify viruses? Or what do we do for virus taxonomy if they're not living things, right? And we talk about, classifying living things according to their evolutionary relationships, and viruses, it's even harder than bacteria sometimes to figure out their evolutionary relationships because you're measuring mutation rates and you're estimating time based on those mutation rates. You can't like say, oh, look, this is a mammoth and this is an elephant and they look kind of similar. Maybe they were related, right? It's not quite as macroscopic as that. So for classification, because viruses have so few things we can identify them by, the classification scheme is pretty simple. We still classify them like they were living things. And I think the highest we go is order. So we still group them, not by kingdom, phylum, class, but we start with order, kingdom, phylum, class, order, family, genus, species. Okay, so that's usually how they're classified, and they're going to be grouped by what they infect. Okay, including all of the viruses that are plant viruses, bacteria, fungus, and protists, and animal viruses. We're up to 63 families and 263 genera is the plural of genus, right? So those are approximate numbers, right? There's more every day. As soon as we find them, they add to the number. So you can see they're not nearly as many as like classifying all of the birds, for example, right? How we classify them is based on very few things. First, on what type of nucleic acid they have. And I'll put in parentheses genome because I use that word over there. All the DNA or all the genetic material that they have. It's like all my markers are running out today. So that's first. The second characteristic that you look for is if they're enveloped or non-enveloped. And the envelope gets precedence. They get classified first, and then non-enveloped And then third is by their size. Okay. Within each genus, and I think it's largest to smallest, I think. And then within each genus, species are numbered. So for example, human herpes virus type 1, we don't really call it type 1 anymore. We just call it HHV-1, HHV-2, and then HHV-3. That's chickenpox shingles, right? So they've been ordered, but that doesn't tell us a lot about their how they infect or what disease they cause. So most viruses kind of still go by two names. They go by their like common name, like chickenpox, and their scientific name, HHV-3. All right, so let's look at a few pictures before we start talking about virus multiplication, because the pictures are really cool. So I want you to see them. Let's see. So okay, so don't look at the words, aren't important. It's like I said, it's for the picture. And I'm actually going to try to zoom the picture even bigger because can I move it around now? Okay, cool. You guys look at E. coli under the microscope and Streptococcus under the microscope, and they're very, very tiny, right? Think about how big Bacillus is and how small relatively E. coli is. You have a hard time telling me if it's a rod or if it's a coccus. So I want you to look at E. coli up there, which is the big pink thing, right? 2 micrometers long. And then I want you to look at all of these, right, all the way down. That's a hemoglobin molecule, just one. That's 15 nanometers. Yellow fever virus, polio is very tiny. Flu, HIV, just pick out some of the ones that you would know, right? Herpes, rabies, right? And notice they're all significantly smaller than the smallest bacteria, right? This is a yeast cell here, which is 7 micrometers in diameter. So very, very, very tiny. So in order to see a virus, we have to use an electron microscope. So, ooh, wait, sorry, go back. So here's some pictures of some viruses. How cool is that, right? So I think the one in purple with the yellow, they've been colored in, right? The computer imaging so that they look like colors. They're all just really black and white pictures, right? I think that one's Ebola, right? B. Yep, that's Ebola because it looks like like a rope, like a twisted rope. A is vaccinia virus. That's what you would get vaccinated against. That's what they use to vaccinate against smallpox. Most of you don't even have to worry about that. And then there's a scanning like image of the smallpox virus. That's letter C there. There's more. Oh, look at this one. This is polio. Each one of these is a polio virus, right? So they're kind of in a lump here. Polio is transmitted fecal to oral transmission. So it's literally, again, from like hygiene, not washing your hands, right? Here's a sort of like a style, an artist rendition of viruses, a couple viruses. These are pictures you're going to see in your book. Here's that capsid protein I was talking about with the spikes sticking out of it, right? We usually kind of just draw it like that. Notice this virus does not have this yellow envelope around it. So this would be non-enveloped or naked virus, and this would be an enveloped virus. Notice this here shape inside, that's the capsid. They're just showing you some different types. This one looks like, this is tobacco mosaic virus. It looks like a stick, right? So they don't always have to be round. Like we get this idea, they all have to look like coronavirus, or they all have to look like HIV. They don't, right? This cool side story, just because I can't help it, even though I didn't have any coffee yet this morning. Cool side story. This is what Rosalind Franklin actually is really famous for, not for her discovery of photo 51, but she actually did X-ray crystallography on the RNA that is inside of tobacco mosaic virus, like right when people were still trying to figure out what viruses were, and demonstrated that TMV had RNA, not DNA, as its genetic material, which is kind of cool. They made a giant model of it and hung it up in the World's Fair. Okay. That's okay. What's this one? I don't know. That's just like an envelope virus. It looks like it has multiple strands of RNA, so I'm guessing that I would guess flu for that one. Flu is probably one of the most studied viruses, like after HIV. Ooh, here's some HIV, I think. Yeah, or herpes. Wait, hepatitis and herpes. What's this one? Don't know. Ooh, there's phage. That's cool. Bacteria virus, right? So we'll spend a lot of time talking about that one because we're going to start with how phage infects and what its life cycle is. I think this is just bigger. I don't know what the one on the left is. I think the one on the right, again, is herpes. That looks like our classic picture of herpes. They've colored the different parts of it to make it more dramatic, right? Like I said, normally this would just be black and white. So look, notice the envelope is rather thick. The capsid is in red here, and herpes virus is a DNA virus. The yellow one, it just says, oh, papillomavirus. So that's the virus that causes warts over there. It kind of looks like a wart. It's kind of funny, but yeah, that's also a DNA virus, I think, papillomavirus. Okay, so just to remind, if the virus is a plus strand RNA virus, right, then that means basically that that piece of messenger RNA can be used immediately to make proteins. If it's a minus, if it's an antisense RNA, then first there's going to have to be some sort of transcription to make messenger RNA that then the cell will use to make virus parts, right? So antisense or negative strand RNA viruses are more difficult or take like 3 minutes longer to multiply inside a host than a positive strand. There's just some different examples to kind of just help you figure out like who goes into which category. I'm not going to test you on this. We're going to talk about these different viruses when we get to the diseases. And then I'll remind you, I'll say, oh, polio is, you know, a single stranded plus strand RNA plus strand virus, or herpes is a double stranded DNA virus. Blah, 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 blah. Okay. That is a terrible picture. It might maybe because I tried to make it bigger. Whoa, okay, no, we don't like that picture. All right, so now we are going to spend some time. I would like it very much if on the handout that I gave you, it starts with multiplication of animal viruses because that's the order they go in your textbook. I would rather us talk about bacteria viruses first and get a good understanding of some of the terms and how they work, and then we'll apply that to animal viruses, all right? So if you're in your book, we're going to flip ahead here to viruses that infect bacteria. And that starts a little bit later in the chapter, like on page 158. And I'm going to start, I think, with letter E there on your paper. Again, so you don't have to write as much. You can just watch or draw the pictures, whatever. We refer to virus, we call it virus multiplication. We usually try to avoid using the word reproduction because viruses don't reproduce, right? They don't have the capacity to do mitosis or meiosis. We wouldn't really call this asexual reproduction. So again, in that vein of trying to remember that viruses are not living things, we change the vocabulary a little bit to help us remember that they're not technically living things, right? So under viral multiplication, then I would like to talk first about bacteriophage. and then we'll talk about animal viruses. Okay, so bacteriophage, as you might suspect, is a virus that infects bacteria. And like other viruses, they are pretty species specific. Like the viruses that infect E. coli pretty much only infect E. coli. They don't infect staph or Pseudomonas or Proteus, right? And the viruses that infect like Streptococcus or Bacillus usually are specific for Bacillus, right? So it is rather species or at least genus specific. You'll see this shortened a lot of time to just phage. Or when I was in graduate school, the snooty people called it phage, right? And that was supposed to be like secret code that you went to UCLA or Harvard, right? Because they said phage, not phage, and everybody else said phage, but whatever. So that's why you'll just see it abbreviated like that in your book. We're going to talk about two different types of bacteriophage or two different types of phage. One group are known as lytic phages. And the reason they're called lytic phages is because they lyse their host at the end, right? They cause a cell explosion, the cell dies, and new viruses are released. The most famous lytic phage is a group of phages that we call the T-even phages, because they were named T2, T4, T6, T8, etc. Right? Their host, the T-even phages, their host is E. coli. So I'm going to be drawing a relatively, bacillus-looking thing for our host and labeling it as E. coli. And I think I'll start drawing over here. I'm going to close that. So here's E. coli, right? Living its E. coli life. Here's the E. coli chromosome. I'm going to make it a different color. Let's make it blue. All right, with its double-stranded, double helix DNA, going along. And along comes bacteriophage. That's specific for E. coli. The bacteriophages, they look like little spaceships. They have this capsid. They have a neck region. It's covered in sheath protein. And they have these tail fibers that stick out. And inside, phage is a double-stranded DNA virus. So, right. Not as much DNA as the whole E. coli chromosome, just a small amount of DNA. And the first thing that has to happen is this phage needs to attach, which we call adsorption. And it does that because, believe it or not, E. coli has little tiny receptors on the surface that the phage tail fibers fit into. So if phage is floating around near an E. coli, it can bind to those receptors. And I'm just going to put on E. coli cell wall to help you, give you something to attach it to. Then the phage does carry an enzyme, and it's lysozyme. And so it releases a little bit of lysozyme, which breaks down peptidoglycan. So the phage attaches or adsorbs, and then it's going to release a little enzyme that's going to start to break down the cell wall. And then the phase 2, or step 2, penetration. The phage injects its DNA like a little tiny syringe into the E. coli. And the empty phage head stays out on the outside of the cell. So I'm just going to leave that like ghost drawing there, and we actually call them ghost heads. So do this. and the DNA comes inside the cell. So now there's a short amount of time where the E. coli, believe it or not, has defenses against small pieces of foreign DNA. And it may enable those defenses, and the DNA may get chopped into pieces and nothing happens. But then that wouldn't be the lytic cycle of phage, right? So we're not going to discuss that right now. Come to genetics, learn about CRISPR, blah, 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 blah. What most frequently happens is the first proteins that are transcribed and translated from the phage DNA are proteins that break the host chromosome into pieces like that and completely disable the host. So even if the host has those defense mechanisms, those CRISPR mechanisms, it can't use them because it can't make them. Only the ones that are still left around. Now remember, the bacteria is full of machinery for replication, transcription, translation. We spent all last week talking about that, right? So there's DNA polymerase in there, there's nucleotides, there's transfer RNAs, there's ribosomes, right? And now the only DNA of interest or available for all of those enzymes is phage DNA, primarily, okay? So step 3, we usually refer to as synthesis. I forgot what they're calling it now. They change the name of it every now and then. We used to call it biosynthesis, but then that sounded too like, you know, now that your book's calling it duplication of phage components. Okay. Synthesis. We don't want to make it sound too much like a living thing, right? So duplication of phage. So let's see, I don't know how much more space I have. I have a little bit down here, so I'll draw this. So the empty phage head is still on the surface. The host chromosome, as we talked about last week with transduction, the host chromosome is over here in little fragments, like not able to do anything, right? The phage DNA, during this phase of synthesis or duplication, the virus has to do 2 things, right? It needs to make or synthesize many copies of the phage genome, right? The virus genome. And the other thing it needs to do is make phage proteins. Looks like my pink is running out. And so that's what happens, right? So we're going to get lots of copies of the pink DNA. I know it's not working very well, but lots of copies of the pink DNA and lots of copies of bacteriophage proteins, some sheath, some spikes or the capsid, some tail fibers, right? Then everything needs to be assembled. Step 4 is assembly. And usually assembly, sometimes you'll see it linked with release, but sometimes not. So inside now the bacteria, all the resources have been used up to produce this little like phage army, you can think of it like, right? Nice little phage army. And then last but not least, because it's a lytic phage, we're going to get release. And that is literally going to be the E. coli is going to be broken open and all of the phage particles are going to be released into the environment. Now, how many phage are we talking about? Well, between 50 and 200 new phage in each E. coli that's infected. That's a lot of phage. 50 to 200 phage. You have to see this picture because it's just really cool. So, real fast. There it is. This is like classic picture. How cool is that? Wait, I forgot to share screens with the Zoom people. So if you're looking at it at home, you won't see it. There we go. Now we both can see it. So this is an E. coli that's been lysed, and these are all the new phage particles that are being released. Notice, this is important for especially us beginning students to remember, the phage doesn't know if the E. coli is dead or not. The phage just has tail fibers that will stick to any receptors on the surface of E. coli. So see all of these phage? They're all trying to infect the dead E. coli cell. Right? They don't have a brain as much as we want to give them, like, you know, brains and abilities to do things that we think they're doing. They don't. They're just a virus. Right? This is a much better drawing of the, you know, the nucleic acid being injected into the bacteria. And this is showing both different types of phages at the same time, which I think is a bad idea. So I'm going to draw you another set of pictures for that. Okay, so. I'll leave this here and try to draw over here so that we can reference back to the other one. So this is a lytic phage. This is a T-even phage. Now I'm going to spend a little time talking about a different type of bacteriophage. It's called a lysogenic phage. The most common lysogenic phage that infects E. coli is one called lambda, which we usually just abbreviate like that. And lambda can do something even more extraordinary than a regular phage. So what lambda phage does is when it attacks the E. coli cell, it still absorbs, still binds to receptors, it still releases some lysozyme. I'm just going to try to draw it a little bit taller or something, right? But when lambda-phage's DNA comes inside the cell, it does this. It forms a small plasmid. And that actually prevents the E. coli defenses that can destroy linear DNA from recognizing it as foreign DNA. Because the E. coli, you can think of it as basically going, oh, wait a minute, is this a plasmid? Has it got something good on it for me? Right? And in those brief moments when it's not breaking this DNA into bits, the phage is starting to copy its DNA, and it's going to start to make, obviously, phage proteins and not bacterial proteins. But when it's in this state, what makes it lysogenic and not lytic is, I should have picked a different color for my DNA. Maybe green will show up a little better. Green. It produces enzymes, the phage DNA, so this came out of the phage, that can integrate the phage DNA into the host chromosome. So the empty phage head is still out there, but now it's integrated into the host chromosome, and what appears like nothing happens, right? The E. coli goes on living its life, and in 20 minutes, as E. coli will do, it's time for E. coli to divide, right? And now we have 2 E. colis with mostly E. coli DNA, but also with phage hanging out inside. And 20 minutes from now, all these cells will replicate, and then we'll have 4, right? And then after an hour, we'll have 8. And then after another 20 minutes, we'll have 16. And suddenly we have this huge population of bacteria that all have phage incorporated into them, like hidden, like a spy, right? This continues until the E. coli gets stressed. Oh, there's when I should use my red. I have a test. I have to work. No, it's usually temperature and availability of water. But temperature stress, right? Water stress. But when the bacteria gets stressed, the phage cuts itself out and goes into a lytic cycle. So it jumps from here right to there and begins madly synthesizing phage DNA, phage proteins, assembling, release, and then the E. coli is dead anyway, right? But temporarily, while the E. coli has the phage incorporated into it, in this situation, we have a name for this phage that's incorporated. We call that a prophage. And that's important because perhaps those of you that know anything about the HIV life cycle can start to see some of the similarities, right? Viruses that cause cancer and viruses like HIV are lysogenic in nature. They go into cells and they can incorporate into the chromosome and become, or into one of your chromosomes, and become a provirus, we call them, right? And they do the same thing. They lay in wait until the cell gets stressed, or maybe until the cell's undergoing mitosis, and then they cut themselves out and cause a lytic infection. Come on, that's kind of cool. You gotta admit, that's cool. All right, blah, 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 blah. Okay, number 3 there, advantages for the host bacterial cell to have a prophage or lysogenic conversion. So you might be thinking, well, why does this phage DNA just stay in there? If it's not causing any damage, you know, can't the host recognize it as foreign DNA and get rid of it? Well, when a bacteria has a prophage incorporated into it, it's resistant to superinfection, which means the phage DNA basically turns off the receptors on the surface of E. coli to bind other phages like lambda. So it's like getting a mini immune system when you have a phage incorporated, well, temporarily, right? Also, the phages, like plasmids, bring bonus genes with them. They often bring a toxin, the DNA to make a toxin that can cause damage to the host cells, which is bad for you if you're the host, but good for the bacteria because it multiplies more, right? When I put toxins, antibiotic resistance, enzymes like, ooh, an enzyme that lyses red blood cells. That'd be handy if I'm a bacteria that doesn't like a lot of oxygen, right? And also, you can gain genes from other host cells via specialized transduction. We'll go back and look at the end of chapter 9. Remember when we talked about sometimes the phage puts the wrong DNA inside? If there's a piece of host chromosome that happens to be the right size, it will go into the phage. And then the next E. coli gets phage DNA instead of, it gets host DNA instead of phage DNA. That's what that means, right? It's usually the same genes because the phage usually goes in that when it forms a prophage, it usually goes in the same part of the E. coli chromosome every time. So when it cuts itself out, it takes a little bit of E. coli DNA and it's usually the same piece and takes it over to the next cell. And then that cell's like, ooh, I didn't have the lac operon before, now I have it. Or, oh, I had that lac operon, now I have a spare copy in case I get some mutation. I can still use lac operon. Okay, so having said that, now flip your page back and we're going to look at some animal viruses and how they infect and how it's different. Again, sorry. like I said, real briefly. So let's do that. Let's see if I can get this picture up here. Yeah, I don't know why it's so wonky. Let's see if I make it a little smaller. Seems to be, there we go. That's the best I can do. Let's look at DNA viruses first. So in animal viruses and plant viruses in general, sort of this is how they multiply. Remember, we don't want to say replicate, it's how they multiply. So you can see behind me, right? So here are, this is inside the nucleus, actually. So I would rather we have this picture first, but I don't know, I guess I don't know why it's so oddly shaped, but it just is. Sorry about that. So the absorption is the same. The virus binds to receptors on the surface of the host cell. The host cell, your cells, bring the virus in. And then what is different is they bring the whole virus in. So with phage, the empty phage proteins stay on the outside of the cell. With an animal virus, the whole virus comes in. So we call that, sometimes you'll see it called endocytosis. Your book is calling it penetration, so it's the same as the phage. And then this is a new step, uncoding, because the whole virus came in and it's literally in a phagocytic vesicle right now, because you're a eukaryote, right? And the phage DNA is not free in the cytoplasm, or it needs to go to the nucleus. It's not in the nucleus yet. So it has to get there, but to get there, it has to get out of the virus protection. It has to get out of the envelope, out of the capsids. So that's the uncoding that happens. So basically your lysosomes and your phagosomes do the virus a favor in that they degrade the protein around the virus DNA so that it can get out and get to the nucleus and infect you. Okay. So we've got adsorption, penetration or endocytosis, and uncoding. And then I'm going to flip over to the DNA virus because this is supposed to be the nucleus, right? So here's the viral DNA coming into the nucleus through a nuclear pore. And then it just acts like DNA, right? It's like, okay, let's start some replication going on here using host DNA polymerase and some transcription that happens inside the nucleus. And then you'll see with some viruses, the viral RNA is going out. We're making virus proteins out here in the endoplasmic reticulum. Some viruses that comes back in and the virus assembles inside the nucleus. Sometimes it assembles in the cytoplasm. It depends on the virus and if it's double-stranded DNA or single-stranded DNA. This part gets more complicated, right? But what I need you to remember is if it's a animal virus and it has DNA, or we look back to this picture, if it has antisense positive strength RNA, some of it is going to go into the nucleus either way to make more of the virus genome. And then other pieces are going to be used out in the cytoplasm to make virus proteins. And then the rest of it's the same. We make the proteins, we make lots of copies of the genome, virus is assembled, and then it has to be released. Two different forms of release, though, in viruses that are, this is a good picture. We have not just lytic release, but also what we call budding. So the virus binds to host receptors. All right. And then the virus, I'm going to say the virus genome or the virus nucleic acid, depending on if it's what type of RNA virus it is, or if it's a DNA virus, that needs to go to the nucleus. And then we're going to have synthesis of viral genome, again, whatever that is, and viral proteins. And then we have assembly. And then release. And the reason I'm writing all this out is because release can either be lytic or, like I said, we call it budding. And there's variations on this, but I love this picture from your book because it's kind of cool, right? So this one is showing you, let's go all the way here, make me smaller out of the way. So this virus is actually assembling on the edge of the cell membrane so that when it buds, it takes some of the host cell membrane with it, which makes it more difficult for your immune system to recognize as a foreign entity, right? Because it looks kind of like you, because it will have some of your proteins on the surface there in that envelope. And then this is a real-time budding of virus where you can see these are mature virions or virus particles, and these are assembling here right on the edge of the new cell membrane, and then being like just going bloop and coming out almost like bubbles. This does not destroy the cell, right? This is great because then the virus just has like a permanent factory, right? Until an immune cell recognizes this cell as infected and gets rid of it, which might take a week or two or never. All right, so last little bit of the chapter, if we flip back to the back side of the page, letter D up there, some examples of what effect do animal viruses have on the actual cells, okay? And your book has this new picture in it, which I'm not crazy about, but this one. So here are some what we call cytopathic effects. This is a giant cell. So what has happened is two neighboring cells have fused together. We call it a syncytium, S-Y-N-C-Y-T-I-U-M. Herpes does this, makes giant cells, lots of nuclei. The cell can't function because it's getting too many signals, then eventually it'll die. Here's a virus that makes inclusion bodies. Those are basically viral particles that are getting assembled inside there. I think this is a picture of cytomegalovirus. So these would be ways we could identify that you had a particular viral infection, right? Cytopathic effects to the cell, the pathology of the cell. Sometimes they also they don't have that picture anymore. Let's see, I'm going to keep scrolling and see if I can find it. There it is. So we also say that viruses cause transformation to cells, meaning make them look different and behave differently. And this is an example on the left there. Those are normal cells. When you put them in cell culture, they grow until they touch each other and then they stop. A cell that's infected with a virus that, let's say, either inhibits mitosis or causes excessive mitosis might make these little clumps of cells here. And the cells lose their ability to know that they're touching each other. And then they form a big pile or a tumor, basically, right? So that can kind of go either way. So I've listed both things there. Cells can be prevented from finishing mitosis, which they might need to do normally. Or if there's loss of contact inhibition, then they do excessive mitosis when they shouldn't. The viral DNA can also go into the host DNA like a prophage and become a provirus. And if it inserts itself near some regulatory genes, near promoters for genes that are supposed to be off and now get turned on, it can cause cancer. Right? So about 10%, 8 to 10% of all cancers are caused by viruses that are, we call them oncogenic viruses, because they cause genetic transformations of cells that permanently alter them. Right. And then some viruses, when they are a lytic virus, how they escape the cell is they break open the lysosome, and then the cell gets basically digested from the inside out, and then the virus can leak out into the rest of the organism. And then I always think it's good to talk a little bit about how we grow viruses in the laboratory. So I listed there as F. So if you're going to grow bacteriophage, basically you have to grow its host, right? So if you're going to grow bacteriophage, you grow bacteria, grow E. coli. We say on a lawn. We usually put it in a like agar, soft agar, and we pour it on top of a plate that's already has some agar on it. So we get this lawn of E. coli, and then we spread the virus over the top. And the virus, when it infects the cells and lyses them, they make clear spots in the media because there's no more E. coli there. Those clear spots are called plaques. That's what PFU stands for, plaque forming units. That's how we count viruses. Plaque, P-L-A-Q-U-E, like plaque on your teeth, right? With animal viruses, there are different ways to grow them. We can grow them in the animal, that's in vivo. Usually people only do that if they're trying to prevent an infection or test a treatment, or they're trying to study the pathogenicity or the course of infection in the animal. Again, because the animals are expensive, they're expensive to feed, they're expensive to keep, they're expensive to buy. They have to be genetically, you can't just like go out and collect 10 rats, you know, they have to be like genetically similar rats, so you have to buy them. More common, people grow viruses in culture, tissue culture, which is what that picture that I showed you before, this picture, right? Where you put the cells in a plastic dish or a plastic vessel, you put liquid media over the top of them, and they grow until they touch each other or whatever they're going to do. And then you can scrape them off and save them. You can freeze them in liquid nitrogen. And then you could use them, to like study, you know, the virus characteristics or how to prevent the virus from spreading from one cell to another, right? You could test new drugs. You can also put in these cell cultures, you can put in immune system cells and document how they affect the virus cells. This is a lot less expensive, more manpower, because you have to, like, there is some babysitting, you know, every day you have to come in and you have to like check your cells, put them in the microscope, look at them, make sure they're healthy, change their food, you know, put them, look at them again. If they're dying or if they get a yeast infection, then you have to do all this other stuff. So it's a lot more sort of daily attention, but it's less expensive than growing the virus in the animal. And then I also put on there embryonated chicken eggs because we still think it's important to talk to you about the flu vaccine, which whatever. So some viruses will only grow in, they'll grow really well in embryonated chicken eggs. So you can forego using the whole animal and just use an embryonated chicken egg. So notice this lab technician here she's injecting virus into chicken eggs, literally. There are different sites to infect that different viruses multiply best. This is how sort of the generic influenza virus is made, vaccine is made, right? Which is why when you get a flu shot, if they ask you if you're allergic to chicken or eggs, you should not lie, because when they manufacture the flu vaccine, you cannot get rid of all of the chicken proteins. So if you're allergic to eggs or chicken, you could have an anaphylactic reaction, right? So that wouldn't be any good. Okay, I think that's enough talk about that.